So spend maybe 30 years now developing just tools to have quantitative measurement at the single cell level. And my interest today is to see, in fact, the correlation between biological function and cell mechanics. And I will give you some different examples. It takes a bit of time because it's, yeah, okay. So this is typically what I'm going to talk about today. So just cantilevers, or yeah, custom-made ones, to quantify single cell here, animal cell mechanics, but I will talk also about plants. And we can couple these two different way of, showing this either just bright field and you can see just the shape of the cell, or we can even go to turf and confocal and have more details and couple mechanics and this kind of action. So I will focus today on the simplest one here, so lateral view, and just mechanics. And when I say mechanics, it's in fact very simple. You have one cantilever, it's rigid, and the other is just a spring, in fact. So you can make very basic uniaxial measurement and force, of course, from deflection. You can get also the speed, we won't talk about it, but you can get the mechanical power, so even you can measure just the energy. The cell is spending to do some work at some point. So everything is in a dish, 37 degrees, and at some point you get the cells, okay? And you get also all these micromanipulators, which like a hooky bat in some sense. I don't go into detail, but this is flat cantilever. You can just catch the cell in between these plates, go up, so you have just one cell isolated between plates. Objective, I won't go into detail also here, but we have this kind of picture, and we can also, and this is an important point, we can at any time get the position or deflection of the probe, so we can have in real time the force applied to the cell, and also the deformation or strain. So we can apply any feedback loop and apply whatever, constant force, constant stiffness, whatever. So anyway, for instance, and I won't go into this today, but I want to show you, we can measure forces, for instance, so single cell just pulling on the cantilever. We can change stiffness and measure adaptation to stiffness in this kind of very simple geometry. And but today I'm gonna focus on the other way around. So applying forces or stress and see how the cell deforms. So just making rheometry, and this is kind of, so we can make it, some oscillation applying, it's lagging, sorry for that, because it's going through the, so you can just apply force, see the deformation, you can just divide force by contact areas, you can get in fact the stress, you can get the strain, deformation, again you can get the modulus with a real part which is the stiffness or elasticity related, and the other part which is the part linked to dissipation or viscosity. And now some examples. So this was a tool. So first example here, so correlation between cell stiffness or modulus and what could be maybe surprising, T-cell activation. So in fact we begin with a T lymphocyte, which is the coordinator, I would say, the principal actor of the immune response. But to activate this, you have antigen presenting cells, many of them, different cells in the blood. And this is like a drawing, and you can see on this drawing that these cells, antigen presenting cells, the ones that are activating T cells, have different shapes, and you see some pictures to show. And at that time, the question from biologists was, okay, maybe these different shapes tell us that there is different mechanics. And could these mechanics, in fact, be important for, so different APC cells have different properties, maybe. And then, if it's true, could that modulate the T cell activation? So, first of all, so you have to measure. So, this was a collaboration with Institut Curie. And what you get monocyte from patient, and biologists know how to put in the solution to differentiate this monocyte into either macrophage or dendritic cells, Second step, and we can measure the stiffness. Second step is to study maybe if inflammation by itself could change the stiffness of these anticoagulant cells. So here also you can either maturate these cells in vivo or in vitro. In that case, we use some drugs here. And then also the question is, do inflammation change this stiffness? So, okay, I'm going to the results straightforward. So here's the kind of map that we found. You can see here. So, vertical scale is a apparent stiffness or modulus, elastic modulus of the cells, single cells. So we begin with the monocyte from the patient. Then you transform this or differentiate this into either macrophage or dendritic cells, and you can see on this scale that in fact you change the apparent stiffness of it. Then also, if you, and this is this color here, if you add LPS, so mimicking bacterial invasion or something like that, you get inflammation, and you can see a macrophage facing this LPS. In fact, you get 1/2, you multiply by 1.5 the stiffness of the macrophage. So you change, in fact, the stiffness of it, and so on and so forth. I want also to mention here that these dendritic cells, when stimulated with TNF-alpha, they are called more or less the best activating cells for the T cells, and you can see it's very, very soft, and it's in fact the elastic modulus is very near the one of the primary T cells. So the question was maybe this very similar elasticities between the T cell and these dendritic cells, maybe it helps activating the cell. And also another thing I want to mention here, you have these Jurkat cells, and these Jurkat cells are in fact model cells that biologists use in lab to mimic the properties, biological properties of T cells. They don't have the same shape, they don't have the same size, but we found that they have almost the same stiffness. So it seemed at that point that stiffness is something that is important in this. So at this point, the question is then, okay, so you have different stiffnesses, and every stiffness of the cell is sensitive to the inflammation, and then could that property just modulate T cell activation? So, yeah, to try to answer this question, what we did first is to have substrate with different stiffnesses, and then with activating antibodies, and put culture T cells on that and measure on one way or another if this stiffness modulates activation. And this is okay. This is more or less the results. So here we're just interested in cytokine production by these T cells, and you have on each graph, so interferon gamma or TNF alpha, and in each case you have plus and minus, and this is about the antibody, anti-CD3. So you are either targeting specifically the T cell or not, from the biological point of view. And what you can see here, in each case, plus or minus, you have these values, in fact, of the stiffness of the gels, so from soft to stiff. And if you don't have specific interaction on the surface, you don't have any effect of stiffness. But once you introduce this specific interaction, then you have a differential cytokine production. So you have a sensitivity. And the other thing you can also see here, so I'm because I want to show you many things. So what that tells us, in fact, is that coupling between chemical and mechanical signal is important for activation of T cells. Sorry. Okay. And also my colleagues have shown, so maybe I have to comment here. So on the vertical axis, you have the different values of the gels over which you have cultured the cells. And here you have the quantity, the number of genes that are either upregulated or downregulated. In the case you have used also anti-CD3. And what you can see, in fact, is as soon as you increase the stiffness of the gen, you increase also the differentiation between upregulated and downregulated. So you have really sensitivity to this stiffness. So this was a first part. So stiffness modulates, in fact, expression. Okay. And then I want to go to another part, and this one more about cancer here. Very quick, and this is collaboration with Saint-Étienne Manville in Institut Pasteur, which is really the one is working on the biological side, because I'm a real physicist, as I mentioned. So here, first, I am comparing the mechanics of primary astrocytes or glioblastoma cell line F98 from rat, and we are comparing them here. And I want to stress here, I'm going to show you that of course you have some dependency of the grade or correlation between the grade of cancer and the stiffness once again of the cells. But here it's going to be more subtle, because a lot of people know that as soon as you have cancer going on, you get softer cells. That's well known. And here what I want to show that we are doing multi-scale measurements. So one here on the left, it's a global measurement, the one with the macroplates. So you have just the whole cell squeezed between these two plates. And here we are looking at the G prime, G double prime, so more or less, let's say, modulus and stiffness. And what you can see, in fact, that okay, you have at this global cell scale a decrease when you go from just primary astrocyte to glioblastoma. Now, if you look with optical tweezers, with speed inside the cell, you look at the intracellular stiffness or viscoelasticity, you see also you only have a decrease. Okay. So you have here everything is softer inside and outside, or whole cell and local cell scale. It's okay. Sorry for that. So now we are comparing human grade 3 astrocytoma and grade 4 glioblastoma here with the cell lines, so with the numbers. And once again, we look at what happens at the whole cell scale, and we found again that increasing the grade, you decrease the apparent stiffness of the cell. But when you go inside the cell, here it's going the reverse way. So it seems more stiffer when you go from grade 3 to grade 4. So it seems that it's not the same. The global cell scale is softer, still softer when you go in increasing grade, but inside it seems stiffer. This is my conclusion. It doesn't, yeah, okay. And we went a little bit further there, so I won't discuss about the cytoskeleton and so on, but maybe the most impressive is what happens at the level of the nucleus here. So using bead inside the cell, what we could do is indenting the nucleus. So we can have a measurement of the either the depth for the force, depth into which we go inside the nucleus, or you can translate this into an apparent stiffness of this nucleus. And what you can see, so here we have the grade 3 in blue and grade 4, and what we end up with, in fact, is that the nucleus is stiffer for grade 4. So the overall cell is softer when you increase the grade, but the nucleus is stiffer when you increase the grade. So this is was something interesting, because a lot of people try to squeeze cells and say, okay, it's softer, stiffer, and try to use this as a diagnosis. But you have to be careful, because if you go very far in squeezing, then you will get to the nucleus, you will sense the nucleus, and then you have the reverse measurement. And Sandrine went a little bit further in the analysis, and here maybe we have to concentrate on this graph here. So they studied, in fact, the relative quantity of lamine A and C, and show, in fact, that the nucleus is stiffer. It corresponds, in fact, to more lamine in the nucleus. So this was a second part, and now maybe a very different one that I wanted to show you, because here you have some like microfluidics like, but maybe strange, I will show you. So here it's a very different system. So now we are in plants, and in these plants we have from the root one specific outgrowth. So this is part of the single cell that is growing and in fact invading the soil. So maybe I will, yeah, so I will comment on this here. So the root and this lateral root hairs, it's really teeny outgrowths, and maybe I can transfer. Okay. So you see here, it's like it's growing. It's a bulge, in fact, and this is used by the cell or the root just to increase the area of exchange to get more nutrient, and also to anchor really the root to make it able to progress in the soil more or less stiff or resisting. And the question, of course, you have a lot of questions over there because now you have drought and you have all the soil becoming more and more stiff, and the question is how this system adapts to this. That's also mechanics, it's interesting for us. Other thing to say, this growth in fact is driven by this very specific high pressure inside coming from the difference in osmotic pressure, like now hydrostatic pressure, and growth in fact is concentrated in the tip. And you have rheometry behind this because you have to control very specifically here the viscoelasticity of the tip to make it just grow the right way and everything. The other thing I want to mention is that while we have another one, yeah. Anyway, so in fact, during growth, and this is in a channel. Yeah, you have the tip growing, but also the nucleus coming from the basis of the cell and migrating from the tip. And it was interesting for us to know if you have obstacles or whatever, what happens to this nucleus, and if it's sensing more or less what's happening, and if you could get a response of the cell to what's happening from the outside. So I will skip this, and we, oops, it takes some time. So you have two kinds of experiment we do with this. So one, you have here the plantum here, so you have the leaves. In this tube, you get the root going on, you have a big channel and you have lateral channels. And this microfluidic, it's not fluid, it's microsolid. So inside, we put in fact agar. So it's first it's warm and it's fluid, and then you get it solidified, and then you can change the stiffness of this agar. And the other technique, and we can combine it both, you can use here again microplates or micro cantilevers to measure forces or apply forces on this and test everything. So I want to make just some pictures. So first of all, this is here. So you have here a central channel. It's too demanding to have it on the same time on Zoom. So you have a central channel with a root coming in here, and lateral channels, and you will see here the root hairs with the nucleus labeled with fluorescence, and they go laterally, and you see the nucleus going inside. So we can follow everything and make some kinetics here with different gels. But as I mentioned, we can also, I won't comment that today, but we can measure here, we can measure here just the force generated by the root hair itself and how it depends this force on the stiffness, for instance, of this plate. Here it's just a zoom, so we can follow, in fact, in the reference of the nucleus. We follow it and see how it deforms. And in fact, sometimes the nucleus is very stiff, but if you put another type here, if you have a kink or change in direction of the channel, what you can see is the whole nucleus is just floppy and bends. So it's really surprising anyway. And this I will comment on. You can just make a deflection here of the root hair, which is more or less a hollow cylinder. And from this simple deflection, you can get in fact the modulus of the wall. So we can have idea about the mechanics and how it grows and everything. So, quickly, sorry. Anyway, oh yeah, let's go. So here, we are just following, in fact, the t, in fact, the length of the root term in time, and we do this for 3 different agar concentrations, so different stiffnesses. And what you can see easily here, of course, is the more stiff, the less the root term is long. It seems more or less obvious. You have also the speed, which is decreasing. And this is in numbers now. So root hair length as function of the stiffness, so more stiff, more concentrated agar, and have less length or shorter root hairs. The same for the average speed or the maximum speed of growth. Everything here is very but more interesting to look. So you have this cylinder with very high pressure, stiff walls. And then what you are looking at, it's not only the position of the tip or the length, but you are looking at the position of the nucleus, the migration of the nucleus. Sorry. Okay. So here you have the tip in black, and what you see, in fact, is that the mean position of the nucleus is more or less following the tip. At some point, it stops and go back. I won't comment on this. And it's interestingly, we can also follow, in fact, the fluctuation of position, because it's not a smooth ride, I would say. It's really fluctuating, but the mean position is following the tip. And what we can see, and it's really impressive, in fact, the fluctuation and also the distance between the nucleus and the tip is changing if you change the external stiffness. And this is something we were not waiting, because, okay, we have high pressure, a lot of things, and how you change this distance and how this distance is sensitive to the mechanical properties of the outside world. And also, I don't show it, but so you have a correlation, in fact, between also the speed at which you grow and the nucleus position. And this we don't really understand yet, but we did a lot of things, but I won't go into details, but we have a lot of things to do here. But at least we show here that the nucleus, in fact, is sensitive to the properties of the outside environment. So then the question is, okay, so if it's sensitive, for instance, could the production of new wall or something like this is changed if you grow in stiffer medium, for instance? And this is my last part. Yes, so we'll just keep this. Yes, so in fact what we do is what I showed you before. So we just apply with the cantilever a deflection to the tube. From this deflection, we can get a kind of force and deflection, please. Okay, so it's very simple mechanics of a tube, but a tube with a very thin shell, so it's very thin. So H here is about 200, 500 nanometers, while R is about 5 microns. So you have simplified expressions. So from F, measurement of F and D deflection, we can get, in fact, an idea about the modulus. And you have here 2 measurements. So one with turbid, so with full pressure, not that. So here I have 2 different stiffnesses, and you have 2 different slopes. Means that you have different stiffnesses when you have grown in different agar gels. And this is the different statistics. Now we have more, but anyway. So you have soft outside gel, soft agar, stiff agar, and what you can see, in fact, maybe it's not something you can worry about, is that in fact the wall is weaker when it has grown in stiffer medium. And with that, I just want to stop here and just so maybe I have a collaboration with my charity, which is a plant biologist, in fact, and also theorist from UK. But I want to mention that we have a lot of young Indian students working on this. And also that David Pereira is the one that did a lot of this measurement on single cent, and Tom Ali also. Unfortunately, David couldn't get a position. This is really bad at CNRS. Yeah, it's really pity because 2 times he was in there. And Thomas Aline is now in England. I finished his PhD and he's a very smart guy. I recommend getting this guy working at some point. Yes, and with that I'm finishing. I just wanted to mention and thank Claire Rose from Asnacios for all the work on the biological side on immune cells. Also Sandrine Etienne-Manville from Institut Pasteur for also the biological part on this cancer cells work. And with that, I thank you for your attention.
Thank you very much, Atef, for this fascinating talk. So time for questions.
Yeah.
Unless you are very hungry.
So while you mentioned of viscoelasticity, all the studies I think that damping part is neglected. Are you separating them, or scale wise it is not that important?
Yeah, so often what we have a power law signature if you look at frequency, for instance. And in that case, what you often see, in fact, that the alpha, so power exponent, is more or less, it's not affected. So it means, in fact, that the ratio between G double prime, so dissipation, and G prime is maintained. So when I say, for instance, elasticity or elastic modulus is dropped by a factor 2, often also the viscous or dissipative part is also divided by a factor 2.
I see. So, but you are calling it a viscometer, rheometer, even for the single cell. So it is sort of nomenclature wise, it's misnomer.
Is that shocking if I say it's a rheometer? If I'm getting G prime and G double prime, it's a rheometer. So please, I don't know.
Yeah, I'll just ask a quick question. So it's very interesting to see, means being a biologist, like when you see someone is working on biology, the perspective that it brings, right?
Yeah.
It's very different. So taking the question from the point of the stiffness of the antigen presenting cell, right? And then you see that the more stiffer cells are better activator for the T cell activations.
The
Stiffer ones are the more better to activate the T cells, right?
Yeah.
Is it because
sorry, I just wanted to mention that in a given range.
In a given
I
range.
showed some range.
Yeah.
But there are people that have studied higher range, and what they claim, I didn't do.
It reduces.
Yeah.
So according to that, it seems to that the dendritic cells and the macrophages will be better to activate the T cell because the data that
just
shows
because of the stiffness.
Yes.
Yeah.
So is it possible that it is because of the dynamics of the receptors and the ligand that basically wants a very optimal stiffness of these complexes, right? And then that makes it to be more optimal to activate the T cell during the state.
So just let me. It's a very important question. It's here. So it's a very important question because, of course, on the gels, the receptors are fixed.
Fixed, yeah.
Yes, and we wondering exactly this value of stiffness. Maybe it's too high, and what does it mean if it's not moving? And what we did, in fact, is we used cells on PTMS gels, and then have the T cells on this, and then we would just we rely on the fact that if you have cells on stiff substrate, it will also be stretched and get more tension in it. And we measured this with specific probe, and in fact we see also that it's more in a more usual range or natural range of stiffness. Yeah, thank you so much. Question.
So I had almost like a similar thing at the end part. That is also the how do you kind of differentiate that or establish the causal relationship here? That's
always difficult to say.
Because
this is for sure. How can I answer this one? This one is very hard.
Because the correlation I can see very strong,
yes,
but how do you
yes,
bring the
yes,
causal
yes.
relationship?
Okay, in that case, I'm not able to answer.
Okay.
For sure. I must say, I have no proof. It happens in other measurements we did at some point. One important thing is the timeline. And in some cases, maybe to 2010 paper we have in the NIS. Here, we did really a change of stiffness in real time.
Huh.
We are able to do that. So mimic stiffness changing in 0.1 second, and then see the response of the cell. So if it's at this time scale, the change means it's mechanical.
Right.
If it's you change mechanics and then you wait for 3 minutes or whatever to have time for signaling, and you show is it rigidity something? It's as the speed is quick as what when you change. So then I would say it's mechanics, and it's I mean the arrow is clear. But here no, I don't have.
In your
similar line,
yes.
yeah, on the same thing, on the similar line, when you are measuring it, what is your outside medium? What is your outside medium? Say, for example, on the cell, say you have done on Viewhurst one and in different grades. So, what is your outside medium?
It's a culture medium.
It's a culture medium,
Yeah.
but if you see in the body, if you change the viscosity of the medium outside, that will have an effect on the stiffness. So when you are commenting that it is stiff or it is softer, so how it is, whether it's a positive correlations or it is
So different questions here. Yeah, okay. So I am measuring on isolated cells.
Yes,
that's
I
for sure.
understand, yeah.
So when it's, for instance, circulating cells, it's okay. When I am using cells, for instance, from endothelium and just isolating them, then it's still maybe going to his question in some
exactly.
sense. So I see even in that state that there is differences and I can scale them, but maybe it's not the absolute value are not the right ones because I am not in the tissue. You're right. But at least I am seeing differences, and it seems to correlate with also activation. But yeah, we have limitation, of course. Yeah, you're right.
And I think I have another question is that in your second part of the talk you measured both G prime and the G double prime, but not in the first part, in the epices. Why? Like you just only talked about G prime and not the
No,
G
we
double
have
prime.
both of them in the paper.
Achha.
But for convenience for this tape or map, we just
But you do, I mean, do you see also a correlation with the G double prime?
yes.
That is what
yes,
you were
yes,
saying.
That was my answer, first answer. Sorry for it.
Achha.
You have this power law signature
Right.
with very little change in alpha, which is the exponent, and means that the ratio between dissipation and storage of energy are
Remaining.
is the same more or less. So changing to
Time
that.
delta is kind of.
The nucleus, how do you imagine the nucleus is sensing the stiffness of the medium? Are there specific sensors that are on the membrane that are directly connected to the nucleus, or there are some other cascade events or
I
something?
don't think so. So I didn't put here, but referees just say, okay, this is speed, but with no explanation what happens in speed. So what we tested, we just played with actin microtubules, as many people do, and we even slow down by playing with the actin monomerization. We can slow down the speed of growth, and you can show in that case that it doesn't affect the nuclear position or very slightly. And this means that it's not something linked to this, at least not signaling whatever.
So there's chemical.
So my point, but we have to prove this, we are working on it. My point is that just when you are facing, so it's trying to grow pressure and you have something resisting, I think that the medium inside is more or less, how do you say, but anyway, more densified in some sense. And so then nucleus is just, because you have a vacuole, which is really a bag with a lot of macromolecules, and it's getting water and inflating, and it's pushing all the cytoplasm. So if you increase pressure, you will in fact push more the nucleus, and you put under more pressure on the nucleus. And to show that, we are just working right now on the diffusion of some particle inside, and shows that when you are, either if you increase osmolarity, so you know that you will make it more compact, you get a slowdown of every movement inside, even the nucleus fluctuation.
And dendritic structure.
And if you put it in a gel very stiff and it's growing in a very stiff cell, we show the same slowdown. But we are working on this. And yeah.
Did you, when you, for the first part, when you compress the cells under the cantilever, and did you go beyond when, do you see any blebbing for, I mean, you know, to have idea about the isotropy of elasticity or
Yeah,
self-extension,
yeah.
whatever?
So,
I mean,
yeah.
do you stop before starting to see some blebbing of the
Okay,
cells?
so blebbing, was a question asked a lot, but I am not able to control it. I mean, I don't have a systematic thing. But if you talk about anisotropy of tension, for instance,
Yeah.
this is very interesting for me. We have now a bioarchive paper, for instance, 1 year. I have a lot of fight with this, but it's another. I have to come back. We can in fact grow cells on micro patterns. We keep everything constant, area, spreading, perimeter, whatever you want, but you change just your aspect ratio, and by mechanical measurement, tension in stress fiber and so on, we can show in fact that you have just because of the shape, tension is not isotropic. So it happens. You can have.
One last question.
Last
Robert.
one. No, so your this route here, that what I found very interesting, because just probably a few weeks ago over coffee I was discussing with my student, just the random discussion, and I had no idea that someone is working in this area. So you have compared soft agar versus stiff agar, but if I put the say particle so that the gel stiffness does not change overall, but the cohesion between the particles are different, so that when the root is growing, it is hitting a stiff, but it can also kind of go through. So I will tell you why I am asking you this question. With the student, we were discussing that how the plant know that I have made a good grip versus not so good grip. How this mechanical crosstalk happens. So
This I don't know. There is Evelyn Kolb in Paris. She's working, okay, not on root hairs, but on roots, going in between these kind of bits. And that's what bits are here functional. So she observed what you want to observe to see how it makes its way in, but also measure forces.
and it must have some kind of feedback loop to tell it that okay, I have
Yeah.
done a good grip or not
Yeah,
yet.
yeah. So on the root hair, and also you have two by archive on it. You have we have in fact using this cantilever, and as I told you, we can, for instance, change the apparent stiffness and change it in real time, and play with this. We are able, or we claim we are able. We see people agree with that, to measure in fact the stiffness and apparent viscosity, and also the how we say le seuil, threshold. Thank you. The threshold to have this growth, and what we are doing right now is playing with this stiffness, and we can show in fact that this, if you change sudden change in stiffness or change in force, we can get these parameters, and we are playing right now with doing this, for instance, with different solarities, or when the root hairs have grown in different gels. So we are trying to know what is adapted in real time on the mechanics of the wall, because this is the one that controls the speed of growth, for instance.
Okay, thank you very much.